In this video we are going to revise the topic B4, Bioenergetics, looking at photosynthesis and respiration, and this is in paper 1 of the AQA Biology GCSE. So we start off with a bit of a recap from B1 really, looking at the structure of the leaf. When we look at the adaptations of a single leaf, it's thin because light can only travel so far through it. It's broad, so it's very wide, large surface area, like we're always saying in biology. Network of veins, and those veins are comprised of the xylem and the phloem, transporting substances throughout the plant. They've got lots of chlorophyll, which is the green pigment found inside chloroplasts, which is what absorbs the light for photosynthesis. Flat, they don't overlap, so you don't see them a leaf directly underneath another one because it wouldn't be able to get any light if it was there. They've got a waxy cuticle on the top of the leaf, and this stops water loss through the top of the leaf. Palisade cells, which are these cells here, we tend to forget this word, but these are the cells that are doing the photosynthesis, kind of our classic plant cell that we learnt at the beginning of learning cells. And we've got these guard cells at the bottom, which they're a profile image at the moment, so they don't look like those kind of kidney bean shapes that you would see if you were looking at them straight on. That stomata, that gap between the guard cells, is primarily for gas exchange, to get oxygen out of the leaf and carbon dioxide into the leaf. At the same time, if you've got holes in a leaf, then you are going to get water loss. Water does not enter through the stomata, okay? It only exits. Up here at the top right corner, we've got kind of just helping us build that word and symbol equation for photosynthesis. So we've got carbon dioxide going in, we've got water going in, and then produced is the oxygen and the glucose. And that needs those the presence of the light and the chlorophyll. They are not reactants, okay? That's why the words are written above and below the arrow on the equation, and we'll look at that shortly. It's kind of like the carbon dioxide and the water is the cake mix, and the light and the chlorophyll is the oven, okay? Without them there, the reaction can't take place, okay? This picture here, we've got a cross section through a stem, and we can see in there the xylem and the phloem. Phloem, as we learnt in B1, carrying the sugar in all locations, so we describe that process as translocation. And the xylem is carrying the water and the minerals, and that's in a single direction from the roots all the way through the stem up to the leaves. And when water escapes through these stomata, we see more water drawn up through the xylem. Okay, just like a straw, as soon as some escapes, more is drawn up through the stem, through from the roots and from the soil. When we look at celery and the stringy bits, that's our xylem and phloem, okay? And the veins on the leaf, that's our xylem and phloem. And we say that photosynthesis is an endothermic reaction because it's using that light energy for the reaction to take place. We would say that the reverse of photosynthesis, which is respiration, is an exothermic reaction, releasing energy, okay? So here we are looking at the word and symbol equations. So we know that photosynthesis is a chemical reaction, takes place in the chloroplasts, and it's where that carbon dioxide and water react together to make the glucose, food for the plant, and oxygen. And it needs that light energy and chlorophyll for that reaction to take place. Now, if we have a question where it says, complete the word equation, don't try and mix it up. Don't write CO2 if it wants words, okay? We must go either full word equation or full symbol equation. And when we look at our symbol equation, carbon dioxide, fairly straightforward, CO2. Water, again, straightforward, H2O. Glucose, a bit more tricky, okay? We've got a sandwich of sixes. So the C6 and the O6 is like the bread. Put those together and you get the filling, which is H12, okay? C6, H12, O6. Yes, you need to memorise that. regardless of what pathway you follow, everybody should know the formula for glucose. Formula for oxygen, obvious one, O2. And to balance it, rather than wasting time in an exam situation balancing an equation, just remember a 6 goes in front of all of the smaller molecules. Okay, so 6, 6 and 6. I won't do working on balancing, we'll just remember that that's what it is. And as I said before, it's the chlorophyll which absorbs the light for photosynthesis to take place. Down here we've got a picture through some cells and you can see the chloroplast there. This leaf here is called a variegated leaf, and this has got some areas around the edge of the leaf that are white. These do not contain any chlorophyll, so there will be less photosynthesis taking place in this leaf compared to a leaf of the same size that is completely green. We come now to our required practical, looking at the rate of photosynthesis. Now we used models, computer models, to simulate this. You might remember using this model down here on the computer where we looked at the volume of oxygen in the, there was a capillary tube here, produced in a fixed period of time. Okay. Equally, we can, and there's another model that does this, we can count bubbles as they're released from the pondweed for a minute. So we can look at volume produced, volume of oxygen produced in a fixed period of time, or we could count bubbles of the oxygen produced in a fixed period of time, probably a smaller period of time if we're counting bubbles. Okay, so we talk about the limiting factors of photosynthesis. When we've got like kind of our maximum rate of photosynthesis, there are no limiting factors. Everything is at its optimum, everything's at its full capacity. But there are some factors that we can adjust to increase that rate of photosynthesis. And you can see here from this first graph, we've got light being a limiting factor at low levels of light. In other words, when we increase light intensity, the rate of photosynthesis increases up to this optimum point. At that point, if you increase the light intensity, it will cost more money for more lighting, but it will not actually improve the rate of photosynthesis. So there'll be a reduction in profit there because money is being wasted on these extra lights or turning the lights up brighter, and there'll be no rate of photosynthesis, no greater production of food, you know, if it was a farmer situation. Okay, so it's a limiting factor until it becomes optimum. When it gets to the optimum, something else becomes the limiting factor. Okay, so if the question says something like, when light is no longer the limiting factor, what else could be? You need to suggest one of the other things. Okay, carbon dioxide, second graph, has the same sort of shape to the graph as light, and carbon dioxide can only be adjusted in a greenhouse, okay, where the carbon dioxide can be contained, or in our reaction down here, when we did our computer model, we used pondweed and we put a chemical in there that increased the carbon dioxide levels in the water. If you've just got the plants outside in the open, the carbon dioxide levels are not going to change enough to make a difference, okay? But same situation, you increase the carbon dioxide, you increase the rate of photosynthesis until you get to the optimum, and then something else is the limiting factor. Temperature all goes down to enzymes and denaturing of enzymes, so we find that the optimum is reached at low temperatures. Particles don't have much energy, so we've got a slow rate of reaction, just like we talk about in chemistry, and as the temperature increases, greater collisions, therefore faster rate of photosynthesis until we get to the optimum. Down this side over here, this is all denaturing of enzymes, wilting of the plant, drying out of the plant, so the optimum has to be at that fixed point at the top. And chlorophyll, well, the more ovens you've got, the more cakes you can bake. So chlorophyll, just you keep increasing it and you keep increasing the rate of photosynthesis. Okay, and the reason that we're measuring down here the oxygen produced is because when we go back to that word equation, we saw that oxygen is a product of photosynthesis. So if we measure the amount of oxygen produced, we can measure the rate of photosynthesis. Lots of oxygen produced, there must have been a fast rate of photosynthesis. And we use pondweed because it's present underwater. We can physically see those bubbles being produced, released from the pondweed, and then collected in the capillary tube, or we just count them. Things that we would keep the same if we were looking at changing light intensity, for example. So we change the lamp here across this ruler, and we'd keep the same piece of pondweed. We'd allow it time to acclimatise to the new situation. So this is quite a popular one. We've got the light being moved closer. We don't immediately then start testing. We need to allow it to get used to the new condition, and then we can start testing. Okay. Similarly, I've seen a question that has different light filters in front of the bulb, so that's another one to consider if they use a different colour bulb. Okay, this is higher only, so it's still double and triple, but just for the higher students. And this is analysing a graph with more than one limiting factor. And this is quite difficult to get our head around, okay, because we can make some basic statements. We can say, if we just look at the solid line at the bottom here, which is low carbon dioxide at 15 degrees, and we compare it to high carbon dioxide at 15 degrees, we can see that if you increase the carbon dioxide, you get a greater rate of photosynthesis. That's one conclusion we could make. If we compare the same concentration of carbon dioxide, but we've got 15 degrees here, we've got 25 degrees C here, we can see that obviously taking the temperature higher has had an impact, a positive impact on the rate of photosynthesis. So we can now say that 4% carbon dioxide rather than 0.03 is better, and we can say that 25 degrees C is better than 15. Okay. Then we've got this situation here where we're looking at light intensity. Now if we look at light intensity on this one here, we can see that the optimum for light intensity at these conditions is about here. However, if we were to look at light intensity first, and we were looking at low concentrations of carbon dioxide and a low temperature, this bottom line, then we find the optimum is actually here. So the importance of what I'm trying to get across here is that even though we might choose one limiting factor at a time to investigate, when we've got through all of them, we haven't necessarily got the ultimate conditions. We have to keep retesting to get the best one. If I'd just done this first experiment with this low level of carbon dioxide and temperature, I'd say, okay, my light intensity optimum is down here. And if I never adjust that again, never check it again, then I'd probably say that the optimum, when I change all the conditions, was here. But if I then find the best carbon dioxide and the best temperature and retest for light intensity, look how far it's shifted, all the way over here. Okay, so we have to keep testing and retesting to try and get the absolute best conditions we can. And that's why that content is higher only, because that's quite tricky to get our head around. Next, we've got the inverse square law, and I've put all these squares here to try and help us understand what's going on. If we move the lamp 3 times further away than it currently is, so imagine we've got our pondweed, we've got our lamp, and then we move the lamp 3 times further away, if we square 3, which is 9, the light intensity will decrease by 9 times. Okay, so moving it 3 times away, so I've got my, this is my pondweed, I've got my lamp here, I move it 3 times further away, 1, 2, 3, it will actually be 9 times less bright. And that's why we've got our kind of times table grid here, okay, because we have to square the number. Okay, so if I move my lamp 6 times further away, it will be 36 times less bright. Okay, you can use that equation as well, but if you just think of it as how many times have I moved it away, square that number, that's how many times less bright it is. Okay, and And this is in arbitrary units, okay? That just means, like, if I said to you, how happy are you right now on a scale of 1 to 5, hopefully you'd say 5, but there's no scale to it, there's no unit to it, so, okay? So arbitrary units just means there is no unit. We see that quite a lot in biology. Okay, so we came across, we've talked about already photosynthesis, making glucose, and we know that that glucose is used as a food for the plant. It actually has 5 uses in the plant. And once upon a time, a few years ago, I was writing this out and I realised I'd spelt the word scarf, which is why we're going to use scarf to remember the uses of glucose from photosynthesis. 1, some of that glucose isn't used at all at that precise moment in time. Now, if we were talking about humans, then we would say the glucose that we don't use is stored as glycogen, in other words, fat. Plants take that glucose and they join it all up into these long molecules called starch. So that is the first S of scarf, it is starch. Glucose being a small soluble molecule and starch being a large insoluble molecule, it builds up into this long chain, and it can be used when the plant has no light, when it cannot photosynthesize, and I tend to call it a bit of a midnight feast. Okay, so it takes that starch that it had in storage that you find in potatoes and all those root vegetables and stuff, and it breaks it back down into glucose. So there's the S. Production of cellulose, and you've probably heard that word before because it strengthens the cell walls. So some of that glucose is used to make cellulose. Production of amino acids. This is a really important one, okay? So photosynthesis, we can't say it directly helps the plant grow, because what it does is the glucose that's made in photosynthesis is then used to produce amino acids, again, a small soluble molecule. When lots of amino acids are joined together, we get our protein. And the protein is used for growth and repair, okay? And we call that protein synthesis. Then we've got our R, which is respiration. Now, all living things respire. So even though the plant is doing photosynthesis most of the time, it's also doing some respiration, just a kind of lesser amount than it's photosynthesizing. So it's using some of that glucose in respiration. And then some of it is stored as fats and oils. Think of like sunflower seeds, think of olive oil, okay, comes from the olives. That's our fats and oils, okay? So we've got starch, cellulose, amino acids, respiration, fats and oils. Okay, that's what we're doing with all of that glucose that's made during photosynthesis. Scarf. Okay, bit of graph analysis here because we know how popular this is in the biology papers. So this is what I'm talking about, saying that plants do more photosynthesis than respiration. This photosynthesis, this is like dawn and dusk, okay? So when there is light, the photosynthesis rate increases. And at this point, most of the day, there is greater photosynthesis than there is respiration. That's why we say the plant's doing more photosynthesis than respiration. See, that respiration level is continuous because respiration isn't down to how much sunlight there is, so it just stays fairly constant. When that dusk occurs and the sun goes down, we see a drop in the photosynthesis. And that links us nicely to this graph over here, because if this was in a greenhouse, we would be able to see a variation in the gases in that greenhouse throughout the day. Let's look firstly at the carbon dioxide line, which is this top line. At night, it increases slightly. That's because respiration, which releases carbon dioxide, is still happening. Photosynthesis has dropped off. So there's going to be an increase in carbon dioxide from that relative increase of respiration. It hasn't actually increased, but compared to photosynthesis, it is higher. In the daytime, we see a lot more photosynthesis. Okay, so therefore, the carbon dioxide levels are going to go down because photosynthesis uses carbon dioxide. Looking at the oxygen line. At night time, that oxygen is reduced. It's being used up in respiration and it's being produced in the daytime because there is extra photosynthesis going on. Now, this next one, whilst it is not a required practical, it is really popular with the examiners. And it's investigating transpiration. Remember, transpiration is the movement of water through the xylem. As water evaporates from the leaves, more water is drawn up through the roots and travels through that xylem, all the way through the roots, through the stem, through to the leaves. And if we put more leaves on this bit of branch here, if it was a bigger branch, we would have more stomata, more gaps for the water to escape. We would see a faster rate of photosynthesis. If we had a hotter temperature, we'd see a faster rate of photosynthesis. The water would evaporate more quickly from the leaves. So what we've got is we've got an air bubble here, and we'd have like a ruler here. And as the water evaporates, just like that straw we talked about, it draws up more water from this beaker and it will move the air bubble along. And from that we can look at the rate of transpiration, how quickly is water evaporating from these leaves. This reservoir here with a tap is to allow some water back in to move the bubble back to its starting position. And this is called a potometer. The other way that they have looked at potometers in the exam questions is they've had a plant pot with a plant in it on some scales with a carrier bag over the soil so that no water can evaporate from the soil. And they look at the reading on the scales, and then as time goes on, maybe 24 hours or 48 hours later, they look at the mass loss on the scales, and then they can see how much water has evaporated from the leaves. And it could only have come from the leaves because there's this bag over the soil to stop any water evaporating from the soil. That would be called a mass potometer, okay? Just looking at water loss. Okay, we had this, another practical that wasn't a required practical, but this was looking at starch in the leaf. So we know that the photosynthesis produces the glucose. The glucose, any excess glucose can be stored as starch. And we know that iodine solution can be used to test for starch. Okay, so in this experiment, at school we had a plant in the light and plant in the dark. The one in the dark would have had that midnight feast, used up all of its starch. So when we tested that leaf, we should have seen no starch. And the one that had been in the light for 24 hours, we should have seen lots of starch produced. Lots of photosynthesis, lots of glucose converted to starch. Now this picture is slightly different. What they've done here is they've just covered up part of the leaf using some black paper. So we've got exposed to light, not exposed to light, exposed to light. We take that leaf and first of all we have to boil it. And what that does is it breaks down the cell walls. And then we put it in some ethanol with the Bunsen turned off because ethanol is highly flammable. Came up on an exam once. And the ethanol removes the chlorophyll from the cells. And this means that when we use the iodine solution on it, we can actually see a better colour change. So we've heated it to break down the cell wall. We've put it in ethanol to remove the chlorophyll. We then put it onto a white tile and we kind of douse it in iodine solution. That just means put lots on, okay? So when we put lots of that iodine solution onto it, we see that where there was sunlight, where it was photosynthesizing, yes, we've got starch, yes, we've got starch. We get that color change from orange to blue-black. In this area where it was in the dark, it's used up, the plant has used up its starch store because it wasn't photosynthesizing, and the iodine remains the orange color. Okay, variegated leaves you can see here, the ones with the white patches, no photosynthesis happening there, therefore no starch in those areas. So similar question, just kind of slightly adjusted. So we're halfway through the topic. This could be a time when you want to have a quick break and come back to it in a moment because we are 24 minutes in. But we're now looking at respiration, okay? And we're going to start off by comparing aerobic respiration and anaerobic respiration. Aerobic respiration is in the presence of oxygen. It's very efficient. Right now, if you're just sitting there, your cells are going through aerobic respiration. It's very efficient, meaning it releases lots of energy and it uses oxygen. Look at the spelling of the word aerobic. It is like aero, as in the chocolate, aero, okay? A lot of people spell this wrong, they miss out that E, okay? So it's aerobic respiration. Again, a chemical reaction. It's the exact reverse of photosynthesis. And then we've got anaerobic respiration. So if we do this constantly, really do it with your hand, stretch it right out and in, you do that for a few seconds and you can feel the pain of the lactic acid building up in your hands, okay? And that is because we've used up all the oxygen, so our cells have gone away from aerobic respiration and into anaerobic respiration. Just in the muscles we're using. We notice it when we run in our legs. We notice it when we carry heavy bags in our arms. So the cells that are used more, have used up that energy, have used up that oxygen, then go into anaerobic respiration. And it only releases a small amount of energy and it doesn't use oxygen. So we've used up all the oxygen that got to our cells. We've still got the glucose, so we're still breaking down the glucose, but just without oxygen, therefore it's less efficient, releasing less energy. And I've mentioned that thing called lactic acid, we'll come back to that in a moment. So we got a bit of a recap to B1 here, where we said, okay, respiration occurs in the mitochondria. These are found floating around in the cytoplasm. Okay, and athletes actually have more mitochondria, so therefore they can do more respiration, they can release more energy, therefore they can be more efficient. Okay, and we've got diffusion in here, so again, recap to B1, where because the carbon dioxide and the water that are produced in respiration are then very high in the cell, they diffuse out of the cell into the blood and then they can be removed by the body. When oxygen and glucose get used up in the cell, they end up being at low concentration because they've all been used by the mitochondria, so then more diffuses in from the bloodstream into the cells. Aerobic respiration as a word equation. Again, reverse of photosynthesis. So we've got glucose plus oxygen. This is our fuel, this is our stored energy. We eat the glucose, or we eat starch, we break it down into glucose, and then we use the glucose for respiration. Plus the oxygen, carbon dioxide, waste product, water, a lot of it wasted. But it's energy that's released from that glucose, and it's how we use that energy. Sometimes you see plus energy, but it's not really a thing, so it's not really a product. Again, simple equation, looks familiar. It looks familiar because it's the reverse of photosynthesis. We've got this glucose in here again. The 6 sandwich with the 12 as maybe the ham in the middle. You could think of it as a ham sandwich if you want. 6, 6, 6 in front of the small molecules for balancing. Now, as we've seen, this is where kind of topics tie together because we've got a bit of B1 in here, we've got a bit of B2 in here. This is the small intestine. This is the villi of the small intestine. Large surface area, thin, only 1 cell thick, large network of blood capillaries, moisture there. All of those things help movement through the villi, through the small intestine wall, into the blood. Alveoli, exactly the same adaptations. Large surface area, thin, only 1 cell thick, large network of blood capillaries, moisture. All of those gases must be dissolved in order to go through from the alveoli into the blood. Exactly the same situation, except this time we're going from the blood to the alveoli. And all of the different ways that we remove water, urine, sweat, and when we exhale. Energy changes, we've talked about this already, so I'll go quickly on this bit. We said that photosynthesis was endothermic. Because this is the reverse, it's an exothermic reaction, releasing energy. How do we use that energy released by respiration? This is directly from the course content. We know how much the course content matches up with the mark scheme. So we've got chemical reactions to build larger molecules. protein synthesis is an example. We've got movement and we've got maintaining body temperature. We're warm-blooded. So when does aerobic respiration stop? Well, I've talked about this a bit already. When we exercise, our body has lots of different ways to try and be as efficient as possible. One, the heart beats faster. Why? Because the blood is the transport system of the body. So if we can move the glucose and the oxygen to the cells more rapidly, then we've got a greater chance of staying in aerobic respiration for longer. Breathing rate. The volume, so the amount we breathe, the deep breaths, and the speed of breathing, the rate, so breathing faster, more breaths per minute, both change. So we increase the depth of our breathing, we increase the speed of our breathing, both to get more oxygen in, to get to those muscles that are using energy, using respiration, and also don't forget to remove that carbon dioxide from the body. When we get to the point where we are doing all of those things and we're still using more energy and we've used up all of the oxygen, we go into anaerobic respiration. This is where the glucose is still there, it's still being broken down to release that energy without oxygen, and it's producing this lactic acid. And we know when we've got lactic acid in our body because we can feel that pain in our muscles. We've said it's inefficient because it doesn't break the glucose down completely. What it actually does is it breaks the glucose in half. So if you imagine that glucose was C6H12O6, lactic acid is half of that. It's C3H6O3. Okay, that's the only thing in this whole video that you don't need to remember. So lactic acid causes that burning sensation when we exercise, and it also results in something called an oxygen debt. When we're in debt, it means we have to pay something back. Okay, so we've got to get the oxygen to break down that lactic acid. Okay, so we do that when we stop exercising. Randomly brought into the course at this point is respiration in yeast. Now, yeast use anaerobic respiration, and we can use this to our advantage because yeast undergoing anaerobic respiration, so just having that glucose, no oxygen, when they break down that glucose, they make ethanol and carbon dioxide. And you'll come across this in chemistry as well. Okay, so we can use this in fermentation to make alcoholic drinks, and we can use yeast in bread making because those bubbles of air holes in the bread is actually the yeast producing the carbon dioxide. Okay, so lots of, well, two uses of it there. You might remember this program we used as well, this computer program here, quite similar to the pondweed experiment. We've got yeast in here. We could change the conditions like temperature, amount of sugar, those sorts of things, and then we could look at how much carbon dioxide was produced in a fixed period of time to, again, compare, so we could find out the limiting factors. We could find out what temperature is best for yeast to respire, for the people who are making bread and making alcoholic drinks to make as much money as possible. Then we go back to exercise, and we've got this graph here where you might remember in class we did some exercise, and then we did the same exercise as somebody else with the same duration of time, the same intensity. Really good example of that would be to use a treadmill at the same intensity for the same amount of time, so you've got comparable results. We can see that the blue line is the fit person. They started off, there's 3 indicators of fitness, and they started off with a lower resting heart rate, indicating they are fitter. When they did that same exercise, their heart rate increased the least. That shows they're fitter. And then the amount of time it takes after exercise for their heart rate to get back to their resting heart rate is called the recovery time. And again, the blue person there has the shorter recovery time. So they have 3 indicators that they are fitter than the red person. Low resting heart rate, low heart rate after exercise, and short recovery time. When we use our muscles lots, they become fatigued, okay? They stop working as effectively, and we really need to rest in order to let them recover. We know that heart rate increases during exercise, and we've talked about why. It's about getting that oxygen and the glucose to the cells. Also removing the carbon dioxide and the water from cells. And we've also talked about breathing rate. Next we'll talk about removing lactic acid. And the lactic acid had that pain in our muscles, and we talked about an oxygen debt. It's the amount of oxygen we need to react with the lactic acid to remove the lactic acid and go back to having carbon dioxide and water. And if you remember, aerobic respiration produced carbon dioxide and water. So I always think of it as a kind of round the houses way of getting to the same point. Okay, so aerobic respiration being the glucose plus the oxygen, making the carbon dioxide and water. And then if you go down the aerobic respiration route, you've got first of all the glucose breaking into lactic acid, and then you've got the second reaction of the lactic acid reacting with the oxygen to get back to those end products of carbon dioxide and water. So the amount of oxygen needed to break down all that lactic acid is called the oxygen debt. Okay, and the one thing just to add to that is that the muscles transport the lactic acid to the liver where this process takes place. And when it says it converts it back to glucose, so it takes 2 bits of lactic acid, puts them together, and you get glucose. It's another way some of it occurs. So some of it's removed one way, some of it's removed another way. Okay, we're nearly at the end now. Okay, we're looking at metabolism. Now metabolism kind of makes more sense once we finish the whole course. It's every single reaction in the body needs energy or releases energy. Respiration is the one that's releasing the energy, giving the energy that all the other reactions in the body then use. Some of that is taking the excess glucose and turning it into glycogen for storage, as we've got here. Some of it is breaking down building up proteins, protein synthesis, which we talk about in Triple Science in paper 2. Okay, so releasing energy, using energy, using energy. And when we really don't have much energy, because we've used all of our stores, we have to break down protein, and this is in extreme situations. Okay, so metabolism is every reaction in the body and how quickly and effectively it happens. Someone with a high metabolism, someone like Michael Phelps, the swimmer, he was eating like 10,000 calories a day in the Olympics because he's got more mitochondria, he's very efficient, he's got a very high metabolism, he was burning so much energy, he had to put more fuel in his body in order to meet his metabolic needs. So we talk about people having a high metabolic rate or a low metabolic rate, and someone with a low metabolic rate could be susceptible to gaining weight, and someone with a high metabolic rate, their body works very efficiently, they burn off that energy, their chemical reactions are happening very effectively. And I think we're down to nearly the very end now. Oh, this is our last slide. Okay, so this is just some, and you could pause this and have a go at these, I'm not going to talk through them. These are some questions that have come from exam papers that you can have a look at answering, maybe talk it through with somebody, some of those answers. Okay, hope you found that useful, thank you.